Hi, this is Miss Litton, and this is my wonderful Period 3 class. Say hi.
Hi.
And we're going to continue on. This is Mendelian Genetics Part 3. Part 3. And we're going to start with pedigrees today. And so this is one example. And when we look at this pedigree, and if I would ask you on a test or on the AP exam, is this an example of an autosomal dominant trait, an autosomal recessive trait, or a sex-linked recessive trait? You would work out all the possibilities. And what would be a good letter to use? A. So if it's an autosomal recessive trait, this daughter would have to be what genotype? Little a, little a. Could the parents have produced that child? What would their genotype be? Big A, little a. So would that work for an autosomal recessive trait? Yes. Would this work for an autosomal dominant trait? No, because if the child had the gene, then they would have at least one big A, right? We don't even care about their second allele, slash or dash. But these parents, neither one of them have that trait, so that would be what? They would have to be what? Little a, little a. So if they're little a, there's no way they could have that child. So we're going to say no to a form of inheritance where it's autosomal dominant. And then we'll talk about sex-linked later, but in this presentation. But just so you know, if it was a little girl, tell me what her genotype would be. XX. Okay, and in order for her to have a sex-linked recessive disease, she would have to have what? Two little a's. Now, her dad up here, assuming that's her father, her dad up here, does he have the disease? No. Okay, so what would it be? X what? Oh, okay, thank you. X big A and Y. Good. And mom would be? That's all we know for sure, right? We don't know her second letter. Right, because it could be masked. If we didn't have any kids, this would be all we know, right? If they didn't have any kids, but here they have this daughter. Well, for her to have two little A's, she could get the little A. If mom has a little A, she could get it from mom. Is there any way she could get it from dad? No, because dad only has big A's. What if, though? Riddle me this. Would it make a difference if mom slept with somebody else?
Yes.
I don't know why. Oh, because I'm doing lines now. Yeah, if mom slept with okay. If mom slept with somebody else, that could be a reason why the trait showed up in the daughter. Do you agree with that? But if she slept with somebody else, okay, that somebody else, what would their genotype have to be? X what? Little a, Y. And then they could have given this little a. Do you agree with that? All right, good. All right, so when you look at autosomal recessive disorders, take a look at this box where it describes them and see if you're comfortable kind of ping-pong back and forth with the characteristics of an autosomal recessive disorder. Go ahead. Okay, can I jump in here on the third bullet? Can you give me an example with eye color for this one? To what parents? Blue-eyed parents will always have a blue-eyed child. Okay, keep going. I'm just making sure I understood that. Okay, let's look at this fourth bullet. An affected individual, remember these are recessive traits. If it's an affected individual, the genotype would be little a, yes? With a homozygous unaffected mate, what would that be? Big A, will have unaffected children. Why? Because the homozygous unaffected mate will always give a big A. Good, all right? Close relatives who reproduce are more likely to have affected children who need counseling, okay? So, because you're more likely to put those alleles together, those recessive traits together. Does that make sense? Okay, and both males and females are affected with equal frequency. So, let me get you caught up there on your notes. So, we were at Mendelian patterns of inheritance in human disease. Okay, and so autosomal patterns of inheritance. So now, I'm not saying whether it's dominant or recessive. I'm just saying it's on the 22 autosomes. Autosomes are any chromosome other than the sex chromosome. Any chromosome other than a sex chromosome. Okay, both males and females are affected with equal frequency. Equal frequency. And we already talked about in a pedigree, circles are females, males are squares. Okay, solid colors has the disease. Autosomal recessive disorders, you already have one. Number 2, heterozygotes have an unaffected phenotype. Here's an example of a recessive disorder, Tay-Sachs disease. This is super sad. Your child is born looks perfectly normal, but as they start to develop, they're doing fine, and then all of a sudden it starts to regress. And they lose the ability, let's say, to control their hands well or to hold their head up. And it ends up in mental deterioration, blindness, paralysis, seizures, and death, usually between the ages of 2 and 5. 4 is very common. We have people within our community, and you can test for Tay-Sachs. There's no cure for it, but you can test for it. All of these children you see in this image have already passed away, because I know when I made this slideshow. So when we have people in our Oak Park community who have Tay-Sachs in their genes, and let me tell you why. It's because of our strong contingency of Jewish community. Because Jews of European descent have a higher percentage of Tay-Sachs disease than non-Jews. Let me tell you why. And this is super sad. During the concentration camps of Hitler, okay, you could be exterminated in concentration camp, or you survive the pathogens, the diseases, the conditions of that concentration camp. If you were a carrier for Tay-Sachs disease, you were more resistant to the pathogens that were in the concentration camps. So this would be a form of artificial selection, where man was selected. So the Jews that survived and lived the atrocities of the concentration camps were more likely to be carriers than the ones who didn't survive. Do you see that? Okay? And so that's why there's a higher percentage of individuals who are Jewish and have Tay-Sachs. And you can test for that, because if both you and your partner are carriers, you wouldn't be alive even at this age if you had Tay-Sachs. But if you were a carrier for it and your partner was a carrier, because you were a good Jew and you married another good Jew, right? And you were a carrier for Tay-Sachs disease, what are the odds that you would have a child with Tay-Sachs? 25%, right? Are you with me on that? Because you could be normal, big N, normal, big N, little n, 50% of the time, and 25% of the time you would be what? Little n, and you would have that disease. Does that make sense to you? There are other examples of that where you see that influence. Sickle cell disease is more likely to happen if you are black than if you are white. And the reason why is, if you are a carrier for sickle cell disease, where your red blood cells sickle, you are more, if you're a carrier, you are more resistant to malaria. So if I do not carry the sickle cell gene at all, I don't run the risk of sickle cell disease, but I could die of what? Malaria. Okay? If I have sickle cell disease, I'm more likely to die of sickle cell disease. And this is an example of what's called codominance, because if you are a carrier for it, then you're not going to die of malaria, nor are you going to die of sickle cell disease. So in Africa, there's a higher percentage of people with the potential of contracting malaria, higher percentage of mosquitoes, the environment, right? And that's why you see that more likely to be in an individual that is black than it is white. Okay? So next, let's look at another disease. Oh, here's Tay-Sachs disease, what it looks like. Cystic fibrosis, we already learned about cystic fibrosis. That is another autosomal recessive disease. Let's take a look at this one right here. Would this work for dominant, recessive, sex-linked? Work that out right now with your bio buddy. Dominant, recessive, sex-linked. Do the work and make sure your eyes are open. Work it out with the letter A's. Okay, so let's start with the first one that we learned, autosomal recessive. If this were an autosomal recessive disease, so I always start with whoever has the disease or the trait. So mom has it, so if it's autosomal recessive, what would it be? Little a, and her husband, little a. Their son does not have the disease, so what would he be? At least a big A. So can this happen? No. So it will not work for an autosomal recessive disease. Okay, what's our next option? Autosomal dominant. So autosomal dominant, that means mom would be big A something and dad would be big A something. Their son does not have the disease, so he would be what? Little a, right? Could that happen? Yes, if mom and dad were both what? Heterozygotes. So would this pedigree work for an autosomal dominant disease? Yes, but not for an autosomal recessive. So what they're going to do on the AP exam is they'll show you a pedigree, and they'll say, based on the form of inheritance that you think this is, what are the likelihood that a child will get it? You see what I'm saying? Or they might say, here's the data. Does this support your null hypothesis? What's your number for it? Why or why not? Okay. Let's do sex-linked. Okay. If this is a sex-linked disease, okay, let's start with the ones who have it. Mom, in order to have a sex-linked disease, would have to be what? X what? Little a. Because she would have to have both of her little a's give her that disease, yes, in order for her to express it. Okay, what about dad? And why? Okay, here they have a son. What is the dad going to be giving the son? He's going to always, there's no bad Y. There are Hollander traits, but not in this situation. So dad's always going to give a Y, right? Mom only can give what? Little a's. So that will not work if it is a sex-linked recessive disease. What if instead of having a little boy, they had a little girl? Okay? If they had a little girl, would this work? No, it wouldn't, because mom always has a bad X to give and dad always has a bad X to give. See how we work that out? Okay. All right. Now, for your autosomal dominant disorders, why don't you ping pong back and forth with these? Go ahead. Does the first bullet point bother you? Affected children will usually have an affected parent. Does that bother you at all? Affected children will usually have, this is an autosomal dominant disorder, so affected children will usually have an affected parent? What do you want to say there? Always, right? Again, this is where I'm going to talk to you about potentially whether a gene is masked or not, penetrance, expressivity, like I talked to you before about polydactyly, and whether or not you express that trait. That's the only reason why there's a usually right there. Okay? Heterozygotes are affected in a dominant trait. Are heterozygotes affected in a recessive trait? No. Okay? Can two affected parents produce an unaffected child? Yes, if they are what for it? Heterozygote. Okay, good. All right. So on your notes for autosomal dominant disorders, affected children will usually have an affected parent. And heterozygotes are affected. Heterozygotes are affected. And two affected parents can produce an unaffected child. Okay, an example of that is neurofibromatosis, having a tan little spot. Okay, if I will tell you at church one time, couple had a baby had a big tan spot on their face. And so I said to the wife, because she was my friend, and I said, Christy, I said, do you have a tan spot like that on your body? And she goes, no, I don't. And because it was a deal, because it was on the face, they needed to have surgery done, etc. And I said, then your husband does. And she goes, how do you know that? I go, I know that. She goes, have you seen it? And it was kind of a joke. And I'm like, no, because if you looked at him, he didn't have it. But the reason was it was on his butt. Okay? So she's like, how do you know? You know, I'm like, oh yeah, I looked. Okay, so the reason is that's an autosomal dominant disorder. So for their baby to have that, one of the two of them had to have that. Does that make sense to you? Okay? I had a student in class who had that covering half their face. Like this, running right down the middle of their face, which affected their vision here on this side of the eye. This is a very difficult thing to carry around your whole life. What can happen is those spots become very worrisome because they can turn into tumors. So that was that picture that I showed before they left. Okay, those spots can turn into tumors. Another autosomal dominant disease is Huntington's disease. Huntington's disease doesn't strike until you're in your 40s usually. So that means you could already have had children, and you could have given your children the Huntington's disease that you're going to develop 10 years later. What's something you could do to help protect against that if you didn't want to pass that on to your children? Not have children. Give me another one, even more obvious. Oh, yes? If you're old enough to have children, what does it say about your parents? They are old. So you would look to see if any of your parents, do they both have to have it? No. Only what? One. Only one has to have it to pass on the Huntington gene that is dominant, right? Now, what if you don't know who your parents are, or one of your parents is? Could that influence it? Yes. You can get tested for these. These are things that you can get tested for, okay, if you think that for some reason it's in your family. Achondroplasia, it's the most common form of dwarfism. That is an autosomal dominant disease, is achondroplasia. So if you have a couple, if this is a dominant disease, and if you have a couple and they have kids, what are the odds that they would have achondroplasia? 100%? No. What if I told you they each had a normal parent? Each of these people had a normal parent. Right? Achondroplasia is dominant, right? So they have at least one big A. But if I told you they each had a normal parent, then each normal parent had to give each of them a what? A little a. Right? So if we did that as a Punnett square, what are the odds that they have a normal child? Yeah, right here, right? 25% that they would have a normal child. All right? People always want to ask me, how do you have a normal child if you're a dwarf and the spacing of the uterus? So let me just answer that. In this form of dwarfism, this part of your body is the same. It's the part that's smaller are your arms and your legs. So your uterus is okay. All right? So did we get all the notes for autosomal dominant? Yes, right? Okay. So for this next pedigree, tell me, do you think this is an autosomal dominant trait, a recessive trait, a sex-linked trait? You ready? Here you go. I'm going to tell you how to look at this. First thing I would assume is, because it's so impactful across this pedigree, I would guess, my first guess would be what? Autosomal dominant. If you only have one choice to pick, and then I would just do the work to see what do you think it is. In this case, I'm asking you, what's the genotype of number 8? First, you would make the assumption it's autosomal dominant, right? And you would have to check it to see if they asked you a question like this. Then how would you find out the genotype of 8? If it is dominant, then you know individual 8 here has to have at least what? One big A. Do you agree with me on that? Okay. So do we know what her second letter is? Yes, you do. Look at her parents. Yeah, it has to be little a. Why does it have to be little a? Number 8's mother doesn't have the disease, so she's little a, little a. Do you know about her father? Do you know her father's genotype? Yes, you do. Yes, you do. The father has to be big A, right? Because he has the disease. But look at his mother. Okay, so what you have to do on pedigrees, do you know the genotypes of their children? What's the genotypes of their children? Has to be big A, little a, because you know they got the big A from their mother, 8. And dad is what? Little a, little a. So you know all of their genotypes. See how this works? Okay, right here, what is this one? What were the odds that they would have 3 children that have this disease? 3 children that have this disease. Every single time this mom's going to give a what? Little a. Okay? Every single time the dad, half the time he could give a big A, half the time he could give a little a, right? So there was a half of a chance here, a half of a chance here, a half of a chance here. 1 times 1 times 1 is 1. 2 times 2 is 4 times 2 is? There was 1/8 of a chance that they would have 3 children all with that disease. Do you see how many different things I could ask you about that one Punnett square or that one pedigree? That's what they could do. They could give you a pedigree and say, how do you think this is inherited? They would ask you questions. What if individual 4 married somebody who didn't have the disease? What are the odds that they would have a diseased child? Okay, none, right? Because individual 4 doesn't have it. Okay, they could ask you statistically, what were the odds they had this many children? If you did a chi-square test on that, does that fall within what you would assume would happen, right? Because we would expect in this situation that half would get the disease and half not, right? And so we got 1, 2, 3 to 4. If I did a chi-square test on that, would that be okay? I bet it would, right? Because it's almost 50-50. But they would maybe ask you what the chi-squared number is. They would give you one that would be easier, right? They would probably have one other child on here or something so it would be even. But if not, you could just do half child. Okay. All right, so those are all the kinds of things with genetics. You can apply it all to pedigrees, and that's why they tend to throw pedigrees at you, because you have to apply all your information on there. All right. Now, So now we are on beyond Mendelian inheritance. And on beyond Mendelian inheritance, we already went over blood types already, right? So sorry if you were not here, but if I was going to simplify multiple alleles for you, in this case, you don't just have big I and little I, but you have big I A, big I B, and little I. And big I A and big I B are both dominant over little I. So then we looked at the different genotypes. This is an example of what's called multiple alleles. This is beyond Mendelian inheritance, right? Because there's more than one way to have the dominant trait. And if you said normally, if I cross 2 heterozygotes, right, we have more options, I guess, on dominance. So you have more options on phenotypes. So on your notes, go to multiple allelic traits to find when a gene has many allelic forms or alternative expressions. Did I already have you take the notes down on that? Oh, okay. When a gene has many allelic forms or alternative expressions. So if I am looking at a homologous pair, I am still diploid right here, okay? But now at this one spot, instead of just having big I and little I have big I A, big I B, and little I. Okay, so blood types are good to show 2 things. One is multiple alleles, because you just don't have big I A and big I B. And the other thing that this is good for is you can show codominance with the AB blood. Okay, so that's why they like to ask blood questions. So you have many allelic forms or alternative expressions. Instead of just 2 choices, as in big A or little a, you could have those 3 options. Codominance is when both dominant allele possibilities are expressed. When both dominant allele possibilities are expressed, which can happen with big I A, big I B, and blood types. Okay, and then I had you cross a type A, type B, heterozygotes, and what you could get. Why don't you do another one for me? Cross a homozygous A with an AB blood, and what do you get? So I'm giving you phenotype, you know, when I say AB, but you would know what the alleles are. I'm just going to give the phenotypes in here. This would be A, this would be A, this would be AB, and this would be AB. Is this what you got? Okay, good. And take a screenshot. I have some practice problems related to that right here. Okay, so codominance is the A allele was expressed and the B allele was expressed, right? So you had AB blood. So in this one, this is incomplete dominance because in this case, your heterozygote has its own unique phenotype. Normally, when we do a monohybrid cross, in your mind, can you cross pea plants that are hybrid for height? 2 of them, are you doing that? So that'd be big T, little t crossed with big T, little t. Yeah? Are you imagining that punnet? Can you smell the punnet? Yes. Okay, so if you did that, you would expect for the genotype ratio to be what? 1, big T, 2, big T, little t, 1, little t, right? Do you need me to do it or can you see it? See it? What would be the phenotype ratio? 3 to 1. 3 tall to 1 short. Look what's going on here. We're crossing 2 hybrids. Okay? R1 is red, R2 is white, and R1 heterozygote is pink. I'm crossing 2 pink flowers. Look, I have 1 R1, 2 R1, 2 R2, right? So what do you give me your genotype and phenotype ratios? They are the same. That's when you know you have incomplete dominance, when your genotype and phenotype ratios are the same, because the heterozygote has its own unique phenotype, right? So the genotype is 1 to 2 to 1, right? If we look at this, 1 to 2 to 1. R1, right? 1, R2. But so is the phenotype. 1 red, 2 pink, 1 white. So if you do a cross and both the parents have the same, let's say, phenotype, but then all of a sudden when you cross them, all of a sudden you have 3 phenotypes, that's a sign, oh, okay, you know, each genotype has its own unique phenotype, and that would be incomplete dominance. If this were codominance, how would this look if this were codominance? I'd still have red flowers. I'd still have white flowers. But what would these flowers look like? Red petals and white petals. Got that? Black bunny versus a white bunny, incomplete dominance. All the hair is gray. Black bunny, white bunny, codominance. I have some black hairs and some white hairs. Truth be told, they're probably one and the same, but the black hair is expressed gene, the white hair is expressed gene as codominance, but it appears to our eyes to be what? Gray, incomplete. But they're probably all about the same. Probably is the same thing. Okay? So on incomplete dominance, did I give you the notes yet? When a heterozygote has an intermediate phenotype between that of either homozygote. Example, red flower, white flowers, pink flower, humans, hypercholesterolemia, and cystic fibrosis. Okay. Here is another example, okay? A person with straight hair, I can always remember the genotype because 1 is straight, H1 is straight, H2, 2s are curly, H2 is curly. A wavy is H1. If I had 2 wavy people have sex, I would expect genotypically and phenotypically to get 1 straight-haired child, 2 wavy-haired child, and 1 curly-haired child. Do you see that? Okay. Now, penetrance and expressivity. I talked to you about that already. Penetrance is, I think you're supposed to have it, but you didn't show up. So maybe it has a 70% penetrance or 80% penetrance on that. Penetrance, complete penetrance is straight up, tall, short, nothing in between, right? Incomplete penetrance is you could have the big T for a tall plant, but you actually still turn up short because you're a heterozygote. So somehow that one T was not expressed. What could affect that? Maybe the environment. I don't know, okay? And then I gave an example with polydactyly would be a good example of that. All right, now, pleiotrophy is one gene giving you multiple traits from that one gene. I would think the best, did I show you this before? Okay, so a really good example of this is Down syndrome, right? Remember the GART gene? That one GART gene, though, there are a lot of things. Mental capacity, right? The digits, the fingers are flattened. The eyes are different. If you have Down syndrome, the tongue is a little bit thicker. So that's a whole syndrome of things that are distinct from each other. Mental capacity and the way your fingers are shaped, two totally different things, but they are as a result of this one gene. That's pleiotrophy. Lots of different parts of your phenotype are impacted. Another example of that is sickle cell disease. You have the two bad genes for sickle cell, but it affects many different things: brain damage, pain, fever, physical weakness, anemia. A lot of different things are impacted by that one gene. Okay, whereas opposed to things that are polygenic, we looked at this. Polygenic is not one gene with a bunch of things, right? It's multiple genes, like A's, B's, C's, D's, all working together to give you that particular, whatever trait you have. And in that, we tend to see a whole range of different possibilities in there. And that's where I talked to you about height. Okay, and could your height be influenced by the environment? Yes. Okay, and I talked to you about nature versus nurture. So if you get good nutrition, you might grow to your full potential of height, but poor nutrition, you could be short. We talked about that being called phenotypic plasticity. Some variables. We talked about twins, whether they ate healthy food or unhealthy food, and if that triggered their obesity gene that they were carrying. PKU, whether you trigger PKU or not, has to do with the food that you eat. If you can avoid phenylalanine as a child, then you will not develop PKU. But if you eat anything with phenylalanine, which there's a lot of food that has phenylalanine, high in phenylalanine, then you will develop PKU and die a very early death. Temperature dependent sex determination. And we looked at polyphenism in insects. Is this an advantage to have your environment affect your offspring? Yeah, because you only want one queen, right? And if you lose your queen, just take one of your larvae and start giving them royal jelly, and you can get a queen out of it. Do you see that? And then insects, some will focus on eating, where others are on sex and dispersal. So you can do the right thing at the right timing for your particular organism. So on your notes, let's hit all of those things. Pleiotropic effects. A single mutant gene affects two or more distinct and seemingly unrelated traits. I gave you examples. Phenotypic plasticity, the ability of one genotype to produce more than one phenotype when exposed to different environments. Height or body composition. Did I give you this one already? Okay. Height or body composition with nutrition. Associated with nutrition. PKU. People who are homozygous cannot break down the amino acid phenylalanine, and it builds up to toxic levels in their body. So a treatment in their environment is give those affected babies and children a diet low in phenylalanine. Temperature-dependent sex determination, TSD, in reptiles, amphibians, and fish, as opposed to GSD, genotypic sex determination. In TSD, environmental temperatures during a critical period of embryonic development determines whether an egg develops as a male or a female. Polyphenism is what we saw here in insects. When the phenomena where two or more distinct phenotypes are produced by the same genotype, major reason for the success of insects, allowing them to partition their life history stages, whether it's feeding and growth or reproduction and dispersal, to adopt different phenotypes that best suit predictable environmental changes, like in the seasons. And number 3, to partition labor within social groups. Polygenic inheritance, when one trait is governed by 2 or more sets of alleles. Dominant alleles have a quantitative effect on the phenotype. It adds to it. The more genes involved, the more bell-shaped curve. Example, skin color, but this trait is also influenced by the environment. How do we know it's influenced by the environment? You can what? Tan, yeah. Environmental influences, multifactorial traits. Traits controlled by polygenes are subject to environmental influences. Things like hypertension, diabetes, schizophrenia, allergies. And we looked at the Himalayan rabbit and how their hair changed. Okay, we talked about epistasis. A gene at one locus alters the effects of a gene at another locus unless you get the gene for hair. If you're going to be bald, it doesn't matter what color your hair is. Same thing here with the dogs. We looked at sex determination. Sorry, we went over this earlier. I'm sorry if I'm going too fast for you. We looked at sex determination is built on the Siri gene. So the Siri gene is the sex-determining region on the Y, the gene on the Y chromosome that controls development. X-linked genes that have nothing to do with gender, yet are carried on the X chromosome. And I told you about Thomas Hunt Morgan and fruit flies, and we looked at some crosses with them. I don't think, the sex-linked problems, I think I went over all of that already. Here, I have some questions for you right here. Oh, yes, I'll go back. You needed something for law of segregation, yes? Okay, you got it. Okay, so going back, law of segregation in summary, each individual has 2 factors for each trait. These factors separate during the formation of gametes. Each gamete contains only 1 factor from each pair of factors. Fertilization gives each new individual 2 factors on homologous pairs. Alleles are defined as alternate forms of a gene. Did I give you that? No. Okay, that occur on the same gene locus on homologous chromosomes. A dominant allele will mask a recessive allele. There are exceptions. Homozygous, big T, little t, both alleles are the same. Heterozygous, big T, little t. Phenotype is the appearance of the genotype. Law of independent assortment, I owe you 1 word there. Each pair of factors segregate or assorts independently of the other parts. Did we get all of Mendel's laws of probability? Chance. Okay, chance has no memory. Each offspring has the same probability of inheriting a trait. It is not dependent on the genetics of the offspring before. Yeah. Wait, guys, I want to hear. Go. Oh, okay. Oh, incomplete penetrance. The dominant trait, not always fully expressed. Not always fully expressed. I will lock my doors if you want to leave your stuff in here for the rally, okay? Have good weekends, okay? And I hope you understand all that.